Hello everyone, we'll just wait a minute or two for some more people to join.
Hi Misha.
Good evening.
Hi.
Hi, Martin.
Hello.
I know it says Diane. That's because Diane and I use the Zoom alternately. And
hi. I wasn't sure. Hi, Derek.
Yeah, just to say, I think we're currently live on YouTube, so we're live streaming this. So if you're, generally speaking, if you keep your cameras off, just to help the smoothness of the Zoom presentation, and then so the talk will be about 30 minutes, and then there'll be a 30-minute discussion afterwards with questions. I think if you have any questions throughout the talk, pop them in the chat, and I can pass those on to Martin. Or if you're happy to talk on YouTube, then feel free to unmute yourself and ask your questions in that way. So I think we've got enough people to start for me to start going on and giving the introductions. So today, yes, this is a deep thought webinar. So before I introduce Martin, I'll just give a few words about the Deep Thought project. It's really about a super collaborative project aiming to bring together researchers from fields as diverse as cosmology, to geology, to astronomy, to biology, to really explore the questions of what are the conditions for life to emerge in the universe, with different properties, with different cosmological parameters, with different charges of the electron, all these kind of things. So perhaps Lucas, if for people who aren't so familiar, if Lucas, you can put the link to the Deep Thought project in the chat so people can explore that in their own time, that would be great. So thank you all for coming. I'm going to introduce Martin. He's a global project leader with a major biopharmaceutical company based in Cambridge. Prior to joining the biopharmaceutical industry, he held a prestigious position as a fellow of the Royal Society at Royal Society fellow at the University of Oxford, working on molecular mechanisms which regulate the control of the HIV-1 gene expression. He has over 200 publications and is a named inventor on 8 patents, and delivered lectures in drug discovery and development, drug delivery, gene therapy, and developmental biology. In 2012, he was elected a Fellow of the Royal Society of Biology for outstanding contribution to science. However, outside of his work in drug discovery and development, he is passionate about all aspects of astronomy, and was elected a Fellow of the Royal Society of Astronomy in 2015 for his services to outreach activities, and has given over 100 presentations, astronomy presentations globally, and contributed to several white papers for the UK Royal Astronomical Society, and is an active science, technology, engineering, and medicine ambassador. He works with local schools to help inspire the next generation of young scientists, and has published 3 papers with young scientists, with a 4th currently under review, I believe. So today is going to talk about Extremophiles, which is the astrobiology side of the Deep Thought project, and why their existence may be supportive of the panspermia hypothesis and suggestive of life being prevalent throughout the universe. So with that introduction out of the way, I'll hand over to Martin for what I'm sure will be a very exciting and engaging talk. So thank you very much. Over to you.
Thank you very much, Joe. And let me just share my screen so that we're good to go. Okay, and we'll put us on screen show. Super. So thank you very much, Joe, and thank you, Lucas, for the kind invitation. And what I would like to do in about 30 minutes is give you a little bit of a tour on extremophiles as a blueprint for universal life, and then leave us with plenty of time for discussion. And as Joe introduced, this is the Deep Thought Scientific Society. So all deep thoughts are encouraged, and there are no such things as silly questions. So for those of you who don't have a biological background or an astrobiological background, please don't feel shy of asking questions and challenging questions, because that's what we're here to do, to learn from each other and to think maybe slightly differently than we've been used to. So just by way of an introduction, what I'm going to take us through is just a few scene setters on life in the universe, then describe to you some of the properties of extremophiles and give some specific examples of classes, and then sow the seeds in your mind about the possibility of some extraterrestrial examples of extremophiles. And then we'll finish up with two particular discussion questions, which, as we said at the outset, is there to provoke some ideas and provoke some questions. So by way of a scene setter, this is something I come back to on a regular basis. For those of you who know Arthur C. Clarke, and hopefully that's most of us, I think for me this quote of his, one of his many quotes, is so poignant. And as you can read, two possibilities exist. Either we are alone in the universe or we are not, and both are equally terrifying. And to an extent, that really does make you think. And it makes you think deep thoughts, because if we are the only life in the universe on Earth, well, we better protect it to start off with. But if we're not, it always begs the question, has life evolved elsewhere within the universe and that we are a recipient of it? Or are there independent evolutionary adaptations of life forms that are present today? It's just that we haven't yet found them. So that's just something to keep in your minds. And then the next one is a wonderful saying, and you'll see that it has been translated, of course, from Metrodorus, the Greek philosopher alive in the 3rd century BC. And this, of course, is translated from the Greek, but I think it sows the seeds, quite literally in this case, about the concept of life elsewhere. And his quote saying, "To consider the Earth as the only populated world in infinite space is so absurd as to assert that in an entire field sown with millet, only one grain will grow." And of course, this is a wonderful illustration of thinking many, many years ago, as you can see, about the possibility of thinking of Fermi's paradox, which is, of course, if there are other life forms, where are they? And the chances are that there should be life elsewhere. But it also gets us thinking again about when we consider our space within the universe, surely it is most unlikely that we are the only life form that exists today. So by way of an introduction, and there's a lot of information on this slide, but it's a really nice publication by Carola Schroeder, published in Chemtex last year, that highlights just about every situation, in fact, every situation that we're aware of on Earth, no matter how the extreme environment is located, where it's located, there are organisms that will grow in it, but also thrive in it. So whether they're at the top of glaciers, at the bottom of the sea, in temperatures in the Arctic or Antarctic, in shallow marine vents, or in ultra high temperature and acidic environments, such as in hot springs or geysers, you can find organisms. And these organisms form into a number of classes. So those that can tolerate very high temperatures, up to 120 degrees centigrade, versus those that can tolerate temperatures less than 0 degrees centigrade. And these are called the hypothermophiles, or the thermophiles, or as we get towards the lower temperatures, the cryophiles. There are organisms that can tolerate extremes of acidity or alkalinity, either acidophiles or alkalophiles. And there are those that can tolerate different salt concentrations that are called the halophiles. And of course, just for completeness here, there are organisms that can tolerate wide extremes in pressures called the barophiles. And those are just some examples, and I'll illustrate some more really specialized extremophiles in a moment. So to give you some pictorial examples, some of these you may be familiar. Oops, what's happened there? The best laid plans. Oh, there we go. Some of these you may be familiar with. In fact, some of these you may have seen. I've seen personally bacterial mats in Yellowstone. So these are organisms that will tolerate extremely high temperatures. These are, of course, in hot springs, but also sulfurous conditions. Black smokers at the bottom of oceans at hydrothermal vents. And organisms that will grow quite happily in pitch. And this is the pitch lake in Trinidad. And the point that I'm trying to make here is that extremophiles are very clever organisms because they adapt to niche environments, and therefore, because they've adapted, acquire a competitive advantage for survival over other organisms that can't inhabit that position. So these are almost, if you like, the ultimate organisms that have adapted to extremely niche environments that outcompete any competition for food, for water, for sunlight, for energy, because there is no competition that can tolerate these environments. So some other examples, and we'll go into one of these in a little bit more detail in a moment. A wonderful beetle at the top there, found in northern Alaska. It does have antifreeze proteins in its blood. So these allow it to be able to tolerate very low temperatures. So this is a cryophile. The organism in the middle, which is a bacterium, Deinococcus radiodurans, which can withstand extremely high doses of ionizing radiation that would certainly kill you or I very quickly. And then endoliths, which are organisms which can live in the rock, certainly up to 3 kilometers and even below the surface of the earth, and live off minerals in the rocks. So of course there's no sunlight, there's very little water, and these are extremely specialized organisms that thrive in these conditions. So some examples of extraterrestrial origin, and this is taken from a publication that Misha Yatsiuta, who's on the call, and I've submitted. And actually, Misha, as of about, where are we? About an hour ago, it's now been accepted. So as soon as I get the proofs, I'll be sending them to you. And that's great news. But some examples here that are now really, really specific. And the two at the top are bacteria that can withstand radiation, one of which I've told you about, which is Deinococcus radiodurans. For those of you not familiar with the unit of radiation here called a gray, 5 to 10 gray will kill a human being. These can withstand doses of 5,000 gray or 15,000 gray with a reduction in viability, or 30,000 gray in the case of the bacterium Thermococcus gamma tolerans, can withstand extremely high doses of radiation. There's another bacterium called Paracoccus denitrificans, which can withstand hypergravity, and it's up to 4,003 627 times G. So 1G, of course, we're familiar with. That's the gravitational field on the Earth, over 403,000 G. And what's interesting is that is the fastest that this particular linear accelerator could generate a G-force at. It is theoretically possible that these bacteria could withstand a greater G-force. It's just we don't have any equipment we can generate a higher G-force with than that. 403,000 G, as I'm sure you can imagine, is some G-force. And there's no doubt that those bacterium thrive under those conditions because of their small size. And that's some very elegant work that was published not so long ago. Other example is one of the tardigrades that can withstand impacts up to 0.9 kilometers per second, so 900 meters per second, which is equivalent to about 1.1, between 1.1 and 1.15 gigapascal shock pressure, an enormous impact pressure. And then last but not least, there is our good friend Deinococcus radiodurans again, or these other two examples of tardigrades that can withstand multiple conditions: extreme cold, dehydration, radiation, acid, exposure to space on the exterior of space vehicles. And they do this by, at least in part, being able to suspend their metabolism to a fraction of the normal metabolic rates. So when we come to think about radiation resistance, I've mentioned Deinococcus radiodurans and the tardigrade. Some very nice work here conducted by the Hashimoto lab, looking at the possibility of now dissecting out what property is it in the extremophile that allows it to withstand this radiation dose. And a paper published, it's 6 years ago now, and it's been replicated by a number of other groups, both in Japan and in the United States, have isolated a protein called damage suppressor, or DSUP. And this protein is able to confer some element of radio resistance on human cells. So the figure on the right-hand side shows what happens to cells that have been irradiated without DSUP, which is the control, or with what's called a sham, a mock DSUP protein that's inactive. And you can see as the radiation increases, the total cell number decreases. As the time increases at a given radiation dose, the cell number decreases. But look what happens to the cells that have got the DSUP protein. Over time, at a 4 gray radiation dose, they're able to tolerate this radiation dose because this protein has been introduced into their cells. So this has led to quite an international effort to develop a roadmap to understand what's the natural predisposition to radio resistance. And this, of course, is interesting and in fact extremely interesting to the medical community, because for patients, of course, who have cancer and who have radiotherapy, there are many instances where cancers are recalcitrant to radiotherapy treatment. And being able to understand what confers radio resistance may have advantage to developing better therapies for cancer treatment, in addition to giving us information on how perhaps to introduce radio resistance where it's needed. So there's the potential of therapeutic intervention. There's the opportunity perhaps for human enhancement at some time in the future. But being able to isolate the biochemistry behind extremophile existence has multiple applications, and I'll give you some examples a little bit later on. So hypergravity, this was published in 2011. So this is a fascinating study with Paracoccus denitrificans, and I mentioned what happens. Let me just turn this off here. Oops, sorry. Turn that off there so I can see the screen better. So this is the profile here of colony forming units, i.e. the number of bacteria per cell over incubation time at various G-forces. And as you can see, as the G-force increases, the number of colony forming units decreases. But even at 403,000 G, this bacterium is quite happily growing up to 50 hours exposure to this level of G-force. It perhaps starts to dip between about 37 hours and 50 hours, but there is certainly a very good growth of bacteria. It's astonishingly high G-force. This is again taken from the same paper, and this shows here, when we look at relative generation time versus acceleration, that the effect of hyperacceleration on the growth was lowest for this bacterium when compared with a number of other organisms. Some of them are bacterium, and of course Saccharomyces cerevisiae is a yeast. So this confers upon us that there is the property within these bacterium that can withstand this ultra high acceleration, hypergravity. And then you have to think, what's the context for that? Are there any conditions on Earth where this would be the case? And the answer is there aren't. There are also no conditions on Earth where organisms are exposed to 15 or 30,000 grain. There are, however, instances outside of the Earth in cosmic explosions, in supernova explosions, where these type of G-forces are believed to occur. So it starts to seed in your mind, why would organisms have this type of ability to adapt to these types of environments when, at least as far as we know, those conditions have never existed on Earth, and they certainly don't exist on Earth today. So I'll give you another example that I mentioned a little bit earlier. So these are the tardigrades. They can survive an impact of nearly, that should say 900 meters per second, apologies, not 800 meters per second. And again, why would organisms do this? What does this mean for us in terms of the possibility of those organisms having come, perhaps from outside of the Earth's environment? So conditions in space. Let's get this back now to thinking about some practical experiments in space. So I think, as we all know and understand, we have low or very low gravity that may cause DNA in cells to mutate. Membrane and 3D organelle structure will be different. And again, for the biologists amongst you, those of you who follow the potential of 3D bioprinting, you'll know that 3D bioprinting now on the International Space Station has been conducted by a number of nations. Started off, I believe, by 3D bioprinting solutions in Moscow. Now some studies have been done by NASA, by ESA, by the Japanese Space Agency. And 3D bioprinting is not an unattractive field because, as in the case of being able to crystallize proteins in a microgravity environment, there is nothing to restrict the three-dimensional growth of an organ or an organoid or an organelle. So the possibility of being able to use the low gravity environment may actually be beneficial, for example, in future possibilities of producing organs for human transplantation. High levels of radiation, of course, are found outside, particularly outside of the Earth's atmosphere, and certainly if we ever enter beyond the solar system into interstellar space, high levels of radiation would be a real issue to contend with. Lack of oxygen, of course, a problem for organs, for microbes that need oxygen, so for aerobes, but perhaps not for anaerobes. And then, of course, extremely cold temperatures and no atmosphere to moderate the temperature. There's a wonderful experiment conducted here by Charles Cackel in his lab at the University of Edinburgh, and this is taken from some time ago. And for those of you who aren't in the UK, Beer, and it is called Beer, it's a genuine name of a village. It's located in Devon on the south coast of the UK. And Charles and his lab took a sample from a rock structure here that you can see in the middle, and they actually applied it to the outside of the International Space Station. And the experiment that they conducted some time ago now was to expose this microbial community in space to high UV radiation, extreme desiccation, and ionizing radiation for about 548 days between February 2008 and September 2009. And they did this specifically to try and explore the limits of life. So with this microbial community, the intent was to ask, first of all, would anything survive? And then if it did, could they identify the species involved? And to cut a long story short, the Space Survivor, and again, I'd urge you to read these publications, and there have been a number since, of course, since this was published, that showed very elegantly that there was a cyanobacterium in this microbial community This was one of the most, it is one of the most primitive cyanobacteria survived and after its 548 days exposure to space. So again, it begs the question, why should a bacterium that's found on Earth be able to survive 548 days exposure to the space environment? What's the evolutionary advantage for that bacterium on Earth to be able to do that when it's not exposed naturally to the space environment? So extremophiles can help humankind. So as I mentioned a little bit earlier about the tardigrades and Deinococcus radiodurans, there are now many, many examples of where extremophiles can help us. And thermophiles can help us in the production of biofuel. Acidophiles, there are a number of examples that can help us in the biomiding and metals and radionuclide removal industries. Halophiles can help us with anti-cancer and as drug leads. And cryophiles can help us, for example, in the production of lactose-free milk. For those of you who use lactose-free milk, you may not know it, but part of the fermentation process is assisted by enzymes that work at 25 degrees rather than 37 degrees, which is more energy efficient and reduces the chance of contamination at the higher temperature. And again, there are also extremophiles that will reduce metals and radionuclides that fall within the cryophile class. So I mentioned a little bit earlier that extremophiles can have some applications in cancer drug discovery. And the molecule on the right here, this is what we call the warhead in drug discovery. And this is an anti-cancer drug discovered from an extremophile. And the drug is now called marisanib, and it's being investigated in a number of, excuse me, in a number of human clinical trials. So we're going to wrap up in a few minutes now, but I just want to summarize and get some thinking within your minds now in preparation for discussion. many examples of extremophiles that you can see in the table at on the top left. In the top right, this is our good friend, the tardigrade. In the middle is a photograph of the Swedish Nobel laureate Svante Arrhenius. And again, for those of you familiar with him, you'll know his achievements in chemistry. But one of the things perhaps he is a little less well known for is that he is believed to be the founding father of the concept of panspermia, which has life existed throughout the universe. His original proposition was propelled on beams of starlight, pressure-driven beams of starlight, which of course we know to be incorrect today. But nevertheless, the concept has life been seeded through the universe, emanating from, whether or not it be from comets or from meteors or from ejection from other planets, the panspermia hypothesis has been really, I think, attributed to him. And then the example on the left-hand side is that the International Space Station and also the photon capsule with ESA and with the Russian Space Agency in the last decade has seen a lot of activity able to conduct experiments for microorganisms in space. So I'm going to finish up with just an example now that'll get you thinking hopefully a little bit more. And that's to introduce the concepts of engineering extremophiles to seed the universe. And it may sound a little far-fetched, but it's based on the fact that there are some studies in place now that are starting to engineer bacteria that can do more than they were originally designed to do or have evolved to do. And this uses the concept of synthetic biology, which is multidisciplinary, biological, bioinformatics, biotechnology, safety assessment and engineering and manufacturing. And it starts to seed in our minds the possibility of making organisms that are designed to do a specific thing. And the possibility of being able to attach those organisms to ultralight nano spacecraft in, for example, the Starshot program, is science fiction today, but may not be science fiction tomorrow or in the years to come. So I'm going to give myself a little bit of a plug for you if my voice doesn't disappear completely. As Joe introduced, I have a real passion for this subject and also to help the next generation. And if you're interested in seeing some of the work that I've conducted in the area, please find me either on Astro Speakers or on LinkedIn. And what I'd like to do now is open up for some questions and to thank you for your time. But to make sure that we have something to think about, ask you first of all, why should organisms be found on Earth which can withstand conditions not found on Earth? So just as a reminder, organisms that can withstand very high doses of radiation, very high G-forces, and impact collisions that sound absurd. 800 meters per second. Why should an organism be able to do that when, as far as we know, none of these conditions exist on Earth? So that's the first question for you. The second question is it or could it ever be ethical to seed the universe with extremophiles, engineered or not? So, for example, those with additional gain of function, natural phenotypes, or could we use synthetic biology created organisms, for example, those that perhaps can metabolize perchlorate, a contaminant found on Mars, and can fix nitrogen? So, Joe, I'll pass back to you, and perhaps what we could do is open a discussion. If I stop sharing, we could perhaps open a discussion. First question is to ask anybody here listening, why do you believe organisms should exist on Earth that don't need the adaptations that they're able to withstand in extreme environments? So what is anybody's thinking?
Well, firstly, thank you, Martin, for a very engaging, very interesting talk. I have many thoughts, some probably more deep than others. So please, please, if you're not comfortable speaking out, using your microphone, feel free to put ideas in the chat, and I can talk through as well. We have Daniele. If you want to unmute. Hello, Daniele. Oh.
Hi. Actually, I was just clapping. It was not a raised hand, but yeah. To answer, yeah. Thanks, Martin, for the great talk.
Thank you.
I'm not really sure. I mean, that's a very tough question. I don't know. I have to think about it. I don't think I got an answer now. I don't know what to think. I mean, many of the things you showed really surprised me. So I'm still processing them.
So maybe I can use my moderator privilege to put in a question / thought maybe. So yeah, one of the things that I was really struck by was that first of your questions of why should it be possible for certain bacteria to be able to exist in conditions that don't exist on Earth. But I wonder if you could say something about what the conditions on Earth have been like throughout its history, especially at the very kind of early stages. And for me, it's certainly plausible for organisms to be able to be very resistant to high levels of radiation, because I imagine that in the very first organisms that existed on Earth would have had to be highly resistant to radiation, and perhaps they are sort of left over from that initial period and are so good at surviving that we're only discovering they're still around now, but may have only developed in the very sort of initial stages. Things like much sort of high levels of G-force is sort of much more difficult to maybe think about how a condition how that could arise on the early Earth. So I don't know, maybe you could say some words about what the conditions are like on the early Earth.
Sure. No, it's a good question, Joe. And I think as far as I'm aware, those levels of radiation have never existed on Earth at the time that there were organisms, that organisms existed either. So if we believe and accept the central dogma of chemical evolution, the timing of it occurred much after the Earth had cooled down, for example, and that the core had formed and what have you. So it would either have to challenge our entire thinking about Earth's formation and the ability not just to withstand high doses of radiation, but ultra high temperatures as well. So that's one thing. And that's a little bit, sort of a little bit of a Weasley answer, because there's no way of confirming it or not. But the question that would come to my mind is, if we believe that adaptation, continuous adaptation, is an evolutionary necessity for organisms to exist, even given the fact that organisms might have existed at the start of Earth's formation, why would they, billions of years later, still have that evolutionary advantage? Why would they retain it? There's no pressure on those organisms to retain the ability to withstand high doses of radiation. And the biology behind it is really interesting, because they don't just have a magical shield around them to protect them. They're as susceptible to radiation as anything else. They just have an astonishingly fast DNA repair mechanism. So their DNA gets damaged the same as any other cells, except the way that they repair their DNA is very, very fast and very efficient. The other property that they have is that they can withstand almost total desiccation. And this is how tardigrades and bacterium survive in space, is because one of the most damaging radiation products of water, the hydroxyl radical, is what does the damage to the DNA. If you don't have any water there, you can't produce the hydroxyl radicals as a radiolysis product of water. So the idea of having desiccation is linked also to the ability to withstand radiation dose. So I think for me, the evolutionary pressure isn't on organisms to be able to keep up this ability to withstand, whether it's radiation, whether it's hypergravity, whether it's impacts. So why do they retain it? And I suppose the same question could be asked as, in that case, does it mean that these organisms are regularly being introduced to Earth? And I don't know that we really know the answer to that, because how would we? We can't possibly monitor every meteorite that hits the Earth, unless there are big ones, of course, and then everybody would know about it, because meteorites hit the Earth every day, at least at a small size. So I think for me, the question would be, what's the advantage to organisms maintaining that ability to adapt when they don't need to? Now, unlike organisms that have developed that ability to adapt because they need to, those that grow in extreme environments, I think the converse is true in this case. But welcome anybody's thoughts. If you'd like to challenge that or don't believe it, or know anything different, please say so.
We have a question from, I know it's not Diane, but...
It's Derek. It's Derek. Thank you, Joe. Thank you, Martin. Fantastic presentation. Thank you. Do you know, has anyone shown that the extremophiles that you described need to invest energy or resources in their individual development as an individual organism? Because for me, that would determine whether or not that extra property, that resistance that you described, is an extreme, is a necessity for its existence, or whether that resistance is a byproduct of some other development. In other words, as it were, an accidental property that it might have.
Yeah. No, I think it's the former, Derek. And I think these organisms have been shown to need and to expend energy to maintain this adaptation. So it does suggest that they're there, it's there for a reason.
That's very convincing indeed, isn't it?
It's very interesting, isn't it? Very, very
compelling argument. Yeah, thank you.
That's a good question, really good question. So perhaps what we could do, Joe, while people are thinking about any further questions on why they're here, maybe think it's a little bit more futuristic, but the idea of synthetic biology isn't something that we'll see in 100 years' time. It's here now. So there are, for example, there is engineered rice. It's called golden rice. It's been around for a long time. It was specifically designed to harbor higher levels of vitamin A for people in the developing world that don't have access to, for example, fresh vegetables. That's why it was introduced. There are many examples of modified foods or genetically modified foods, as was called in the 80s and 90s, that are, again, products of synthetic biology. So that's here now. We can do that now. We can modify organisms to be able to work in extremes that aren't natural, if you like. What's the audience's thinking about not just our ability to do it, because we can do it, but whether we should send those organisms deliberately out into space? Because it could be done. Is it ethical? Does it serve a purpose? Or is it just a nice scientific experiment? Let's see what happens and worry about the consequences later. What would we think? Because if they sow the seeds for potential life in future, and perhaps if we put it in the context of Mars, everybody's thinking about Mars at the moment. If we could seed Mars with extremophiles, is it an ethical thing to do?
He's got his hand up, so...
Yeah. I mean, I don't see any, well, maybe I have to think about it a bit more, but I don't see the ethical problem, you know. So here on Earth, we generally value preserving life. So why is there an ethical issue when we try to preserve life outside Earth, right? But maybe this definitely requires more thought. But this is my initial.
So one thought to have in that is maybe an analogy, which I don't know whether it's appropriate or not, but you know, when a new animal or something is released onto an island that has never seen it. So, you know, I know cats in, I'm no ecologist, but, you know, cats in New Zealand, for example, or rats, or on an island with ground nesting birds, and absolutely so decimates the local ecology. I don't know if it's even, you know, appropriate to assign words like ecology to extremophiles, but you know, there may be extremophiles out there that if we sort of release different our own sort of favorite extremophiles into space, then I don't know whether they will compete in some sort of extraterrestrial ecosystem, and that might have impacts there. And then should we care about that? That's a thought that came to mind. But anyway, we have Misha. Misha has a hand up. Okay, do you want to unmute?
Yeah, I'm not sure about this one, but from what I'm aware, NASA goes to quite a lot of efforts to sterilize some of the stuff we send to Mars, and I think it was on the orbit of the gas giants in order to not to hit Europe and other places where life could possibly exist, since if we introduce our life to there, if there is life, it could be completely wiped out by our life forms. So I think that's the ethical issue.
Yep, you're absolutely right, Misha. And this is one of the biggest issues within space ethics at the moment, which is that fear almost of contaminating another world. And it then gets into the ethics of considering, do we as human beings have the right to contaminate another world? So it becomes even deeper than our first set of deep thoughts. So yeah, you're absolutely right that the space agencies go to extraordinary lengths to make sure that there is the minimum chance of introduction of contamination. So for example, the lunar landings, the Martian rover landings, there will be others hopefully in the future. but there is a lot of effort that goes into that for precisely the reasons that you say. And the reason I asked the question for everybody is that particularly on Mars, as you may know, one of the principal blockers towards building a sustainable supply chain for food on Mars is the nature of Martian regolith. So Martian regolith has a contaminant in it called perchlorate, and perchlorate is effectively poisonous to the growth of crops. And it doesn't do human beings much good either, but it will stop crops growing. So perchlorate is also a byproduct of the munitions business on Earth. And there are bacterium that have evolved to metabolize perchlorate, and they're found in those regions on Earth. So it's not impossible to introduce perchlorate-eating bacteria, if you like, into Mars, or in a limited way into a Martian environment. But if we're thinking about converting Martian regolith to being something that can sustain crop growth, we want to do a little bit more than that. And the possibility exists to be able to engineer bacteria that can both metabolize perchlorate, but also fix nitrogen, and give us the possibility there of increasing regolith consistency and quality. So that's a real conundrum, and it's something that the space agencies are thinking about now, because when and if we ever get human beings to Mars, they'll need to find a way to sustain their lifestyle, sustain a colony. And of course, it begs the question, which is exactly Misha's point, which is it right to be able to introduce a modified bacterium that we wouldn't even have on Earth into a Martian environment where we don't yet know whether or not there is life on Mars or has been, even in an extant form. So that's something else to ponder about if you can't sleep at night.
Wow.
It may even help you sleep at night, I'm not sure.
Yeah, no, plenty to think about there. Daniele, you've got your hand up.
Yeah, thanks. I think what I was thinking was related also to what you, Joe, was saying earlier. So I would say my first reaction was to the question was also like, if you have any evidence that there are other kind of bacteria, let's say on a planet, then you would need to be very careful to introduce something else. But even if you do not have any evidence, like that doesn't mean that there could not be other bacteria which you didn't detect yet. So I guess that probably in this case, in any case, the most ethical choice would be perhaps the conservative choice of just not do anything, because you never know. On the other hand, as Martin was mentioning earlier, the possibility of colonization of other planets from ourselves, we would be contaminating them anyway, not with engineered bacteria, but with whatever we bring with ourselves. Yep. So maybe, I guess, if that's something that we want to do anyway, to try and colonize other planets, maybe the question about introducing engineered bacteria or not could become less relevant. I don't know, or maybe I'm missing something.
No, it's an interesting point, because I think this is the real dilemma that faces, I think, those that set space policy and those that work in the ethics area related to space, because they have this all the time. And of course, this is one of the reasons why, and I forget the vehicle that Misha was talking about that was deliberately crashed some time ago to avoid.
Cassini.
That's right, Cassini. Perfect. Thanks, Misha. That was deliberately crashed to prevent the possibility of contamination. So it was a very real, and is a very real, ethical dilemma for the space agencies. Well, plenty of deep thoughts there, Joe.
Yeah, plenty of deep thought. Yeah, I mean, arguments for and against colonization of outer space. Yeah. Angela Nicholson has her hand up. The question on that, Nick.
Hi there. It's we're sharing a Zoom call, so it's Keith here from the Caithness Astronomy Group, and I mean, I think this is a really interesting discussion about ethics, which is really appropriate to have. I think my concern is the increasing involvement of the private industry, the private sector in space, and whether they share those same ethics, or whether they have other priorities, and how we might give some governance and control on the irresponsible use of space. We've seen that already to some extent, but I'm a bit concerned about just, whether it's SpaceX or others going to planets for commercial gain with less interest in the ethics of ensuring that the spacecraft are not contaminating the planets.
No, again, it's a really good point, Keith, because I think if we were to be perhaps a little bit cynical, let's think about what's the value in being able to land an orbiter on a comet or a meteor. It's scientifically fascinating and challenging, but actually I wonder whether or not the purpose is to think about extracting minerals and rare metals from those bodies and bringing them back to Earth. So I'm sure that there is an agenda, and personally, I would like to see the agenda open because I think that gets the public behind it. It also gets the scientific community behind it, and it gets funding behind it. But I think there is some way to go yet before we have a truly open discussion about the interests of private enterprise, because there has to be a return on it. I don't think anybody would believe or anybody would expect that private enterprise is prepared to spend probably trillions of dollars on a space program and not have some form of recoup on that investment. But I agree with you entirely. I think there needs to be regulation, but there also needs to be a very open dialogue about the possibilities of being able to mine, for example, minerals on the moon, minerals on Mars, minerals on comets, because you can see that that's a direction that as soon as it becomes cost effective, you would bet that that's where we'll go.
I do feel, Martin, that whether it's a private enterprise or a national enterprise, there seems to be such a very strong push to achieve colonization of Mars that, and as you said earlier, the perchlorate in the regolith is a major obstacle. If the deployment of extremophiles is actually a clear answer to that perchlorate, I personally think there will be so much pressure to achieve the colonization that the ethical question will actually take a back seat. And that's more of a reflection of politics, human nature, and human development than anything else. It's certainly unlikely to be given a sort of a pure treatment and a pure answer to be returned. More likely that political and other pressures will actually prevail.
Yeah, I'm sure you're right, Derek. And also there'll be no doubt some competition between nations wanting to do that first and best and what have you. Yeah. As we see today, it does make you again think a little bit about, well, we've got everybody queuing up at the moment to land on Mars and explore Mars. I'm not sure how much collaboration there is in advance of making sure that missions build on each other rather than try and replicate what each other's done.
Any questions? If not, I might just ask one question, again related to I guess more ethical questions. But there were two words in that talk which really caught my attention, which were human enhancement. So how I mean, we're not talking about, you know, making the Avengers, right? But, you know, how realistic a possibility is it to take biological adaptations of extremophiles, such as, you know, high radiation, being able to, you know, survive under high G, yeah, high pressures example, and you know, confer those abilities to humans per se, as you were saying. There's a paper that seemed to be doing more or less exactly that, and what that kind of what that would say would be useful for medical purposes, but I also imagine that would be useful for, you know, human exploration in space as well.
Yeah, it's a very good question, John. It's something that's actually really close to my heart too, and I think there's a number of ways of looking at it. So in the first instance, non-biological human enhancement is actually, it's been around for centuries. So if we think of something as, in fact, I can see a number of people wearing enhancements already, because if you're wearing spectacles, then you've got a human enhancement already. If you have a hearing aid or an organ transplant, then that is also a human enhancement. So I think those have been around for some time. I think if we take it to the next level, the concept now of brain-computer interfaces has been around with increasing success and complexity over at least 15 years. Now at the point where, as we've probably all seen, the possibility now of patients who have tetraplegia, for example, to be able to communicate in ways that would have been thought laughable only 10, 15 years ago. So that's developing. The prospect of being able to develop perhaps even extrasensory perception or telepathic skills has been around for some time. And there was in fact, there still is a project that the Defense Agency in the United States is funding, Silent Talk, that is there for the military, but is there, if I remember correctly, there is a device that can associate your thoughts with your brain patterns and then code those in a way that can send them to a recipient, such that that recipient can receive those and then deconvolute them and understand what the signals are. That's the theory behind it. It's not gone, as far as I'm aware, or actually as far as in the public domain, beyond that as a concept. But you can see that that's the way that that field is going. And as we start to think more about the possibility of, it is, if you like, it's probably in a way almost the ultimate upgrade for human beings. Is there the possibility of starting to merge biofabrication with human physiology? I'm sure that that will happen. So that's one track. The other track is gaining a lot of credibility, but not for the reasons that we're thinking about just now. And that's gaining credibility from the gene editing field and gene therapy field. So again, for people with very rare diseases on Earth, gene therapy is now much more of an accepted procedure than it was 20 years ago. I used to work in the gene therapy field, and the field virtually stopped around the world because of an experiment that was conducted in the late 1980s that showed it, at least, to be a very unsafe thing to do. Regulation has got a lot tighter now. There are gene therapy products available. Gene editing is at a similar stage in its existence, which is that there are clinical trials underway, and some will read out, I think they will read out at the end of this year, where the possibility of now being able to edit somebody's genes who have a defect for a particular disease. And it's a disease that I can never remember the full name of, but it's LCA10, which is a hereditary form of blindness. There are some clinical studies that are underway to be able to edit the gene that should, in theory, be able to restore, partially restore the sight of patients who are losing their eyesight, or if not restore it, at least stop further loss of their eyesight. If those studies are positive, they open up a vast field of which ethics has to be a part, because it sows the seeds in our minds, again, not just about the prevention of human disease or the prevention of worsening of human disease, but the possibility of being able to augment our natural genetics, which could induce perhaps the protective effects of radiation, possibilities of withstanding microgravity so as not to lose hard or soft tissue mass in the way that astronauts do in microgravity. And there are a whole series of other potential benefits of being able to engineer DNA. But again, if you think about the ethics of it, let's just say, for example, I could be made radio resistant if I wanted to be, and if there was the justification for doing it. How would you ever be able to ethically justify making people radio resistant so that they can go into space? Well, how would you test it? How would you test, has Martin become radio resistant? We'll give him a high dose of radiation. Oh dear, no, he isn't, he's dead. So, I mean, that's an extreme, but you can see what I mean. How would you conduct a clinical trial to show that somebody has become radio resistant? You could take some of their cells ex vivo and radiate them and see what happens. But actually, that's not getting to the central point. So I think the whole field of human enhancement, at least at the genetic level, in a way that's not elective, i.e. isn't necessary, is a very, very ethically contentious area, and one that will be debated, and one that will, I'm sure, be brought to a head. I would predict over the next 5 years, because we have the technology to be able to do it. So a very infamous study, and it's given a little bit of a bad press because it's always blamed at China, but it's not something that I think is specific to China, because I think there are other countries doing these types of studies, that twins were gene edited to be deficient or to be defective in what's called the chemokine receptor 5 target, which is it's a receptor on the surface of cells, and it's actually a co-receptor for HIV-1. And the idea was that you would ablate this receptor, so you would, in theory, make these twins not susceptible to HIV infection. Now, that was done. That was done throughout any ethical consideration, as far as we know, which was why there was a great deal of concern in the scientific community. But what we don't know, of course, is CCR5 as a receptor for HIV isn't just there to be a receptor for HIV. It has other functions. Like everything else in our bodies, they don't just do one thing. Mostly, they don't just do one thing. So the idea that we could pick and choose almost what we would want to permanently ablate or delete needs a lot of ethical thought before it happens. So I think that's a very long-winded answer to your question, Joe, but there's an enormous amount of work being conducted in the field of human enhancement. And it ranges from the concept of seeing Terminator 3s wandering around the planet, which of course sells books and sells films, and actually are quite good as well, if I'm completely honest, down to the more mundane perhaps, which is to think at a very localized level, but very importantly, at a medical level, what could be achieved for the benefit of people who aren't in space in the first instance, and then how could it be transposed to those that want to travel into space?
Well, fantastic. Thank you for that answer, Martin. We are, I think we're pretty much out of time, but I'm sure if you have any questions, I'm sure you can email, I'm sure you can send Martin an email and he might be able to respond. So I guess before we wrap up, I say I hope you enjoyed this, this Deep Thought webinar. This Deep Thought YouTube channel. There's a Twitter, Facebook account. So if social media, please feel free to follow along, and you can be notified through there about any future events that we'll be holding. So I think with that, we'll call it an evening, and I hope you go away thinking many a deep thought. So thank you very much, and thank you especially to Martin for what was a really fascinating talk. Let's give him a round of applause. Yeah.